All right, we have done a lot of metabolics. Now we're going to talk about anaerobic versions of metabolics. So, some anaerobic respiration and some fermentation. So, this is what happens when you don't have oxygen. Now, oxygen makes the best, the most efficient, with the least interesting and toxic waste products energy. So, it is used by most things on the planet. But there are plenty of things that live in places without oxygen for some reason or another. And so there are things that have developed lots of other ways to do stuff. Now, most of these things are single celled organisms. And in fact, a vast majority of them, almost all of them, are bacteria and archaea. Essentially, the thing that makes eukaryotes is that they picked up other little things. And one of the big things they picked up were mitochondria. So they've had mitochondria for a very, very long time. So the vast majority of them are aerobic. But these cute little anaerobic bacteria are probably some of my favorite things. So let's have a quick discussion about them. So, anaerobic respiration implies that we're going to go through the entire process of respiration. So that includes the citric acid cycle or a version of it, and also the electron transport chain or a version of it. Now, if you'll remember, at the end of the electron transport chain, the goal is to capture the electrons so they don't go floating all over and causing trouble. And so we put them on an oxygen, O2, oxygen gas molecule. That is our electron acceptor. Other things can be electron acceptors. It doesn't have to be oxygen, and it can produce some very interesting things. All it needs to be is something that is in an oxidized form and therefore able to be reduced. So O2 to H2O is one of the most obvious of those, but here are a number of others that you may or may not have thought of. So let me tell you a little bit about Geobacter. Geobacter is a genera of bacteria that are found in soils, and not just like the soils at your feet, mostly deep soils, soils where you have to dig down a good distance and then you are getting pretty far away from oxygen. And they are neat little buggers. I spent a number of years in a lab studying them. And so this one here, I believe, is a Geobacter sulfur reducens, which you can already hear a little bit about what it does, because if it's sulfur reducens, it might reduce sulfur. There's also a metalli reducens, which reduces metals. And the really neat thing is these little blue bits here are pili that it actually can use to transfer its electrons. Talk more about that in a moment. The other neat thing is that although I did not take this electron microscope image, and I do know how to do that, but I didn't in this case, I did Photoshop these really cool colors in. All right. So metalli reducens, it can reduce a metal. In this case, the biggest metal that it reduces is iron, which is really neat because what this bacteria can do is it reduces iron 3 plus to iron 2 plus. So again, reducing electron acceptor. So it's adding electrons to it. And what that does to iron is it takes it from the iron oxide form, which is the one we associate with rust, remember? So oxidized iron is rusty, and sends it back to the mineral or metal type form. And in fact, in some cases, not every strain does this, but in the ones that do, it actually sets the iron up in a way that makes it already reactive to magnets. So it creates sort of a magnetic sludge along with it and can actually produce the stone or mineral magnetite, which is an iron sulfur complex that we use to make magnets. It's all about the electrons. So isn't that neat? The other neat thing about these bacteria, Geobacter, is that in place of iron, they can reduce uranium. And uranium is a naturally occurring metal. Of course, we overdid some areas of uranium by mining it and then taking out the part we wanted and leaving a lot of leftover uranium. And so what you find is Geobacter grow in these areas that we cleaned up mostly by capping them over, but left a lot of uranium in the ground. And one of the things you can do with the Geobacter is activate them by giving them something to eat. Usually you would give them an organic acid of some kind. They would take the uranium and they would reduce it. And the difference between oxidized uranium and reduced uranium, oxidized uranium travels in the groundwater and was getting kind of all over the place. Reduced uranium actually sticks in the soil and then can stay sequestered a little bit better. So that was the goal of that project. It got a little crazy though, and I'll just tell you a little bit why in a few of these future slides. But Geobacter are neat bacteria. Another thing bacteria can do is reduce nitrate. This is not entirely uncommon, especially in water systems. So nitrate is NO3. And when you reduce it, so again, in reduction, we're going to either take away oxygen, add hydrogen. We're going to add some hydrogen to this. You can get as far as, and this takes a couple of steps in some cases, NH4, which is ammonium, and in water systems, sort of ammonia. So if you've ever been by a lake, and if you live where I do in upstate New York, you've probably been by a lake. In summer, the lakes settle out in such a way so that all the plant matter falls to the bottom where it's kind of dark. It gets respired, it uses up a lot of oxygen, and it leaves a lot of bacteria that can reduce other substances, including NO3, so nitrate, and sulfur. And it reduces the nitrate to ammonia and the sulfur to sulfhydride. Ammonia, you probably know what that smells like. Sulfhydride is the thing that makes the rotten egg smell. Then in the fall, the temperature of the water shifts. It causes the lake to turn over. The water moves from the bottom and it's able to actually move back to the top. And it releases ammonia gas and sulfhydryl gas. And so if you are driving by a lake in early fall in upstate New York and you smell rotten eggs, that was anaerobic respiring bacteria at the bottom of that lake turning over. So here's the sulfate ones. You can see SO4 is the sulfate, and in that form, it's just some stuff in rock. You're not going to notice it, but sulfide is this black, icky stuff that has the very strong rotten egg smell. It does, in fact, turn all of the sediments black as well. This was part of our problem with the uranium reducing, is that there was a lot of sulfate in that system. And so if we were able to activate uranium-reducing bacteria, we also activated sulfate-reducing bacteria, and suddenly ended up with a lot of hydrogen sulfide, which is not much better than uranium. So in terms of toxicity, still a problem. However, interestingly enough, the hot springs out in that part of Colorado also very much were sulfur smelling, and all of us thought they smelled just fine after doing our field work. All right, so here's our lake turnover where you can see pre-turnover, the bottom portion is able to separate out, and then afterwards, all of it comes up, and so it releases the ammonia and the sulfide gas. Another thing we found along with the uranium, because it is again a natural metal, and the two tended to associate with arsenic, and it turns out bacteria can reduce arsenic too. So if they were able to reduce the uranium, some of them were also able to reduce arsenic. It's amazing what bacteria and archaea can live on. They practically can do anything. Strongly advise taking a microbiology class. All right, so this is another one of my favorites because methanogens are typically archaea, so a little older and a little sturdier than bacteria, that use carbon dioxide as their electron acceptor, which is kind of interesting, and it reduces it to methane. So methanogen because it produces methane. So here's the really neat thing about these bacteria. They very often use CO2 for literally everything, like absolutely everything. They're able to break the CO2 apart to start the energy production process. They're able to use the CO2 to capture the electrons at the end of the process, and they're able to take some energy from it and use that to create the organic molecules they need to build themselves with. These are archaea that can live almost exclusively on CO2. And like a few other things that they need. They need micronutrients and phosphates and nitrates and stuff like that. Got to have DNA and RNA. But most of their structure is related to CO2. And what's really interesting about that is that that's mostly because, and you think of CO2 as a gas, but they actually live in rocks because CO2 interacts with some water systems in such a way to make CO3, which interacts in such a way to make various kinds of limestone. So most limestones are actually carbonate type of minerals and rocks, and therefore we're talking about archaea that live in rocks and produce methane. Some of these also live in the bottom of wetlands, so where it gets very, very organic, and therefore, again, respiration uses up the oxygen, you get methane, and then the methane bubbles up, and your wetlands are all methane producing. It's kind of neat. All right, so the very last thing of the anaerobic respiration is what can we do with this kind of stuff? Especially, so this goes back to the Geobacter that have those little pili that are able to pop their electrons on things. Now, obviously, most of them are popping their electrons on things like little iron atoms that are hanging out around them in the rocks. But what if you could get them to drop their electrons on something else? Specifically, to drop their electrons on something that we could make a circuit out of, like a real circuit, not an electron transport circuit, and use to actually light something. That's exactly what these are. So these boxes, inside of these boxes, are essentially a carbon sheet. Typically, sometimes they're a platinum sheet, and they're dropped in the bottom of water. So they sink down to the bottom where there's very little oxygen. The bacteria that can do this job, that can do anaerobic respiration, will grow on them, and they will use them as electron acceptors. They'll drop their electrons onto the large carbon sheets. The carbon sheets are able to pick them up and send them along the wires, and sends them along the wires in such a way, in this instance, that it lights the light on this buoy. So it is a self-lit buoy, and it doesn't rely on solar energy like a lot of things do. It relies only on the energy picked up from the bacteria at the bottom of where it is. So it's settled into one place, and it can use that energy to go. Another version of this that people that I knew had worked on was a sort of latrine version, where you dump your electron accepting sheets in the bottom of where you are collecting your human waste products, which definitely go anaerobic pretty quickly. And then you create a circuit that uses them to light the light in the bathroom. Isn't that neat? So there are some versions of this being done in areas probably that are doing things, it's not quite composting, but if you've talked to people who are trying to use that kind of thing to create energy, this is usually what they're doing, and they are relying on the electron accepting capabilities of bacteria who typically use metals, like Geobacter. So I think that's neat. All right, gear shift here to fermentation. So the previous processes assumed you were going through, again, all 4 steps of cellular respiration. But if there's not an electron acceptor around that you can use, you don't want to let those electrons go flying. Again, they're very dangerous. That's why you don't stick your hands in sockets. Electrons flying around are problematic. So most biological systems have a way to stop the process, essentially at pyruvate. So at the end of glycolysis, the system will say, well, is there an electron acceptor available? If there is, we get to move forward. If there's not, we stop immediately at the end of glycolysis, except we have a small problem at the end of glycolysis. We created some energy, but we turned all of our NAD plus into NADH, and we're stuck. We can't make any more energy until we actually reoxidize the NADHs. They're holding on to energy to use later, but we could keep making these little ATPs if we could just loop them back around. So is there a way to do that? Well, it turns out there is, and it's not an overly difficult way, that the energy from NADH can be used to make a very slight change to this oxygen on pyruvate, break that double bond and turn it into an H and OH, which actually creates lactate. Now, of course, here's the thing with lactate. Lactate is also in water systems known as lactic acid, and if it stays in our systems, and especially other large multicellular systems, it does damage to local molecules. So that can be an issue. So we can use lactic acid fermentation for a short period of time, but we really need oxygen to come in, turn the system on, turn the lactate back into pyruvate, and get it to go down the line. But what if you're a tiny single-celled organism that, who cares if you produce a little bit of this acid? You're just going to spit it out and move away from it. Lactic acid fermentation bacteria do just that. rather than dealing with an electron acceptor, in many cases, they either are bad at using them or they, for some reason, don't have one available, will go ahead and be like, well, I can just keep creating this little bit of energy. I can make lactate while reviving my NAD, and I can just spit it out. So who cares? Why spend all the extra energy on this process? And that's what they do. And in fact, if you want to find the tiny single-celled organisms that do this, you want to check your yogurt, because this is the process that produces yogurt. They start with lactose, which is half glucose and galactose, and you can turn galactose into glucose. So they start with lactose. They're able to create the pyruvate. Then they go ahead and create the lactate. They spit out the lactic acid into the milk product. So the sugar gets eaten. The acid goes into the remaining whey protein. And interestingly enough, what it does is it interacts in a way that solidifies it into yogurt. That's how you get lactic acid in yogurt. Same thing happens in sourdoughs and in sauerkrauts. So anything that really produces something sour is probably a lactic acid fermentation process from bacteria. There are a lot of different fermentation processes because bacteria can actually turn pyruvate into a number of different possible molecules, again, as long as it can spit them out again. But there is one more that is of interest to us as biologists and human beings. This one is more common in some yeasts. So although yeasts are eukaryotes, they do have some real interesting things they can do that make them like single-cell organisms. And then some bacteria do this as well. In that, you take the pyruvate, and actually they allow it to do the first step, which is the pyruvate decarboxylase. So it goes ahead and it pops off that CO2. Requires a few extra pieces, but that's fine. And then it has one more enzyme that does the switch from the NADH to the NAD+. So it starts with the acetaldehyde. So you remember we end up with an acetyl group after pyruvate oxidation. In this case, it's not attached to a CoA, so we just call it an acetaldehyde. We hit that with an alcohol dehydrogenase. It's able to add, in this case, so this is a dehydrogenase. It would take a hydrogen off an alcohol, but it adds one to the acetaldehyde, creating ethanol. And this process is probably best known in things like beer and wine production, actually beer and champagne production, because for both of those, what makes this process work really, really well is that you cap it and you capture the CO2. CO2 makes the bubbles. The ethanol makes the alcohol, and now you have a bubbly alcoholic beverage, all thanks to the microbes using fermentation. Isn't that neat? Or you could have them do this process, let the CO2 bubble off on its own, capture the ethanol, and do other things with it, such as distill it, collect it. All of these do the same thing. All right, so that is all the cool stuff that some bacteria and archaea can do without oxygen. And some of it even makes very interesting food items that you can use. Thanks.